I think it's very hard to put into words what the impact of Dr. Arnold O. Beckman is on my life, because the magnitude of his impact is so large. Oftentimes I find myself crediting Arnold Beckman with the foundation of the field of biochemistry. When I teach biochemistry, I tell my students that biochemistry is the study of how thousands of different biomolecules come together and give rise to life. We mainly talk about 4 different classes. We talk about proteins, nucleic acids, lipids, and also sugars. It turns out that we know a lot about these biomolecules because of Arnold Beckman and because of his invention of the DU spectrophotometer. Arnold Beckman contributed the invention of a brilliant set of tools. He contributed the DU spectrophotometer, he contributed the pH meter, he also contributed the ultracentrifuge. These inventions have had and continue to have a large impact on the fields of medicine, agriculture, and industry. But how does this relate to biomolecules? The first publication that used the DU spectrophotometer was about one member of the broad class of lipid biomolecules. And this member is vitamin A. And because of the DU spectrophotometer and because of the characteristic light-absorbing properties of vitamin A, scientists were able to determine its concentration in different nutritional supplements. The spectrophotometer played a foundational role in early biochemistry, but we still use it all of the time today. We use it in my laboratory to measure the concentration of proteins. We also use it in our lab to support our Arnold and Mabel Beckman Foundation research on photosynthetic chlorophyll pigments. Receiving the Beckman Young Investigator Award early on in my career gave me confidence. It allowed me to pursue fundamental science but ask high-risk and high-impact questions. The award has also prepared me for success because it's immersed me in a community of people that learn and abide by Arnold Beckman's rules for success. My favorite rule is there's no satisfactory substitute for excellence. My lab studies photosynthesis because it is arguably the most important biological process on Earth. It is a process that organisms use to convert light energy into chemical energy. It's a process that liberates the oxygen that we breathe, and it's a process that fixes CO2 into the sugars that we eat. So my lab is really interested in studying photosynthesis because it's important for life. We also study photosynthesis because it's adaptable. So one way that photosynthetic organisms adapt to environments with different colors of light is they perform chemistry on the scaffolds of their light-absorbing pigment. So the most abundant light-absorbing photosynthetic pigment in nature is chlorophyll A. What's remarkable about chlorophyll A is different derivatives of it are found all throughout nature. These derivatives differ by the addition or replacement of some functional group on the photosynthetic scaffold. What's amazing is that these seemingly simple modifications actually have profound influences on the wavelength of light that a photosynthetic pigment absorbs. So when I look at all of the different chlorophyll derivatives, I actually think about them as evolutionary adaptations to available light. It's very difficult to functionalize the CH bonds found on the chlorophyll scaffold. Now, in biology, the way that photosynthetic organisms perform this chemistry is they use metalloenzymes. But metalloenzymes aren't always the easiest to work with. They're oxygen sensitive. They also use a lot of enzymes that are embedded in the membranes, and that means that they're poorly soluble. Now, very similarly, when we talk about chlorophyll as a substrate and chlorophyll as a product, it's also poorly soluble. It's also oxygen sensitive, and it's also light sensitive. There's not a lot of information available on how the different pathways are used to make photosynthetic pigments. So oftentimes trying to study these pathways outside of the cell is limited by a lack of knowledge of all of the proteins and all of the molecules that are needed for the chemistry to occur. So from the very beginning, when I started my laboratory, one of the things I understood is that to ask the questions we were interested in asking, I would need a very interdisciplinary group of scientists. We use structural biology, we use cell biology, we use biochemical techniques. We're also able to harness inorganic, organic, and analytical methodology to do our science. Our overarching goals are to gain an understanding of how photosynthetic organisms exist in diverse habitats. Our long-term goal is to leverage this information and overcome current hurdles in agriculture, industry, and medicine. So, With the support of the Arnold and Mabel Beckman Foundation, to date, my laboratory has been able to isolate and study a protein that is involved in making the photosynthetic pigment chlorophyll B. This work has been very exciting. We have been able to demonstrate that through combination of this protein with two other partner proteins, that challenging chemistry can be performed on the chlorophyll scaffold, and that a direct precursor to chlorophyll B can be generated in a test tube. We have also been able to use this cell-free biocatalytic system to access small amounts of a non-natural chlorophyll pigment precursor. Because chlorophyll B has a formal group rather than a methyl group on its pigment scaffold, its absorbance spectrum is blue shifted relative to its precursor chlorophyll A. Now, additional formulated chlorophyll pigments also exist in nature. These pigments, namely chlorophyll D and chlorophyll F, however, unlike chlorophyll B, show enhanced absorbance features in red and far red light. Now, collectively, due to their altered spectral properties, formulated chlorophyll pigments have received a lot of attention. They've received attention for their potential uses in medicine, and they've also received attention for their potential uses in agriculture. Unfortunately, however, making formulated chlorophyll pigments is challenging, so their chemical synthesis requires more than 46 different steps. In addition, the proteins involved in making these red-shifted chlorophyll pigments are currently inaccessible to study outside of the cell. Luckily, now that we have the protein involved in making chlorophyll B, we can work to engineer it to put that formal group modification at different positions on the pigment scaffold. In doing so, in the future, we would like to generate a library of non-native or native formulated chlorophyll pigments that are currently inaccessible. In addition, through formulating chlorophyll pigments and changing the wavelength of light that they can absorb, we hope to ask the questions, can we engineer crops to grow in the shade of another crop? Can we continue to use nature's solutions to enhance food production? Can we pursue alternative energy solutions? Can we contribute to greener ways to making chemicals and biofuels? Can we engineer plants to use all available sunlight? And can we engineer photosynthetic organisms to grow irrespective of environmental conditions? Because of the support of the Arnold and Mabel Beckman Foundation, we've been able to ask bigger questions and think outside of the box, and how we can take what we find in my lab and try to apply it to a lot of different disciplines.